Hi there, I'm 12.Wirthlin. I'm a neuroscientist at Carnegie Mellon University, and I'm interested in the evolution of behavior. And one of my favorite behaviors to study, of course, is Manakin courtship displays. How these incredible birds have evolved to be able to do all the incredible, highly skilled motor behaviors that are a part of these elaborate courtship displays that you've heard about today. And so if we want to understand the evolution of courtship displays, or really any complex trait or behavior, we need to be able to connect the genomes of these species to the behavior in some way. So if we look at all the genomic resources we have available, on the one hand, we have the genomes themselves. And so in this talk, I'm going to be focusing on 5 Manakin genomes that we've sequenced as a part of these efforts. And this gives us really just the raw genetic material. And a genome on its own is sort of just inert. When things get interesting is when a genome turns into a transcriptome. So a transcriptome is the picture of all of the genes that are active in a given tissue at a given point in development, in a different behavioral context, and so forth. And luckily with Manakins, we have a number of transcriptomic resources available, ranging from muscle tissues in the golden-collared Manakins to areas of the brain involved in social behavior in the wire-tailed Manakins, as well as several other tissues from various other species. Where I come in and where my interest really is in the epigenomes. And so when we talk about epigenomics, we're talking about everything sort of above the level of the genome, different modifications to the genome that allow for gene regulation to take place, which is how you get these tissue and behavior and time point specific transcriptomes out of the genome. And again, the two main regulatory regions involved with this are enhancers and promoters. Promoters are directly upstream of genes and generally are less cell type or behaviorally context specific. And really it's the enhancers that are guiding the highly specific tissue and contextually specific gene expression that we're interested in when we're talking about phenotypic evolution. And so mechanistically, how this happens is through changes in genomic conformation. And so this has to do with various ways the genome can be methylated, or more commonly, how histones can be modified in different biochemical ways to either close chromatin up so that gene expression is suppressed, or to open the chromatin up so that gene expression and gene regulation can take place. And this epigenomic regulation is really what gives us, which takes us from an inert genome to all the tissue and cell type specific expression that gives us a full animal, with these highly specialized tissues and cell types for instance, all the behaviors that we're really interested in when we're talking about Manakin courtship displays. And it's not just the patterning of, a human baby or a Manakin, but rather many different complex traits that we know have evolved through genome sequence evolution, specifically in enhancer sequences. So for instance, there have been enhancers that have been connected to the evolution of the human forebrain relative to chimpanzees. There have been enhancers connected to limb loss in snakes, as well as to the loss of eyes and vision in subterranean mammals. So clearly, gene regulation and enhancers is really a critical hotbed for phenotypic evolution. And so again, just to sort of take the broad picture here, epigenomics is the study of this dark matter of the genome. Whereas the protein coding gene sequences that most of us think about when we first start thinking about connecting genomes to phenotypes, those only take up about 2% of the genome. And these non-coding sequences are the other 98% of the genome that we're still actively trying to figure out exactly what it does. And perhaps, again, the most interesting and most relevant sequences within the non-coding genome from the perspective of trait evolution are these enhancers that regulate gene expression. In the human genome, we have over 1 million of them. They take up a much larger proportion of the genome than genes. And mind you, there's something like 20,000, give or take, genes in the human genome. So this is many, many enhancers regulating each gene. So this is why you can have the same gene being redeployed in different behavioral contexts, in different points in development. And these regulatory regions evolve much more rapidly than genes do, which is why this is, again, considered to be a likely source for phenotypic evolution. Some of these genes, if you tweak them, you can disrupt a whole suite of processes. But because these enhancers are so cell type and tissue specific, and depending on endocrinological conditions, behavioral conditions, they're so highly specific that they can be tweaked in all kinds of interesting ways, much more flexibly than gene sequences themselves. So this is what we really want to find. We want to find, for instance, an enhancer that has some gene sequence change between mannequins that perform these incredible courtship dances and non-mannequins that don't. And we'd love to find a specific set of enhancers that lead to changes in gene expression in an interesting part of the brain that could control courtship dance behavior. And this, we might posit, could be really the, potentially a causal mechanism for how this behavior evolved. This is what we'd like to find. So how can we get there? Identifying regulatory enhancers is not trivial. If we want to look for genes, we have all kinds of signatures, in terms of, looking for codons, intron, exon boundaries, all sorts of signatures to allow us to pretty efficiently predict genes in a genome, as well as being able to take genes from nearby species and map them onto a newly sequenced genome in order to annotate that genome. That's much trickier with non-coding regulatory regions because, again, they evolve so quickly and we don't have the sequence language of them quite figured out yet to be able to predict them efficiently. So here are options, roughly. We can either experimentally determine them, for instance, with this attack-seq method, where we identify all of the regions of the genome that are open and available for transcription factors to bind. And the pros of an experimental technique like this is that we know exactly what tissue we collected, what behavioral condition, what sex, what age, etc., that we collected this tissue from. And so we have a high degree of specificity in terms of knowing exactly what context and tissue that set of enhancers was involved in. The con is that these experiments can be somewhat difficult to do. They require typically very fresh tissue or well-preserved tissue that's processed quickly in a fully equipped molecular biology laboratory. So that can be tricky to do with these birds that largely live in remote field conditions. The other option we have is to try to make predictions based on sequence conservation. So the pro of this approach is that it only requires a genome to be able to identify these regions. all we do is sort of subtract out the coding regions, which are of course highly conserved across species, and try to look for these little spots like this that are in a non-coding region but still have roughly high sequence conservation. The cons here is this is not tissue specific. This is just, the raw genome. So we have to make predictions about, what tissue we hypothesize it might be involved in, or we'll have to do some follow-up, which is what I'll show you a little later, how we can do some of that. We are also going to miss those more rapidly evolving species, clade-specific type regulatory enhancers that may be involved in, subtler, more recent tweaks to a phenotype. But these two approaches can be highly complementary. Certainly with the experimental approach, we can't profile every single tissue, every single behavioral and developmental condition. So again, I think, both of these are valid approaches, but this is the approach that I'm going to be talking about today. So we do have some helpful resources if we want to make these computational predictions. One is UCNeBase, which is a database of these ultra-conserved non-coding elements. And these are likely enhancers that are conserved all the way from human over to fish, with 95% sequence identity. And they're also, of a reasonable size to be a regulatory element, over 200 base pairs. So this database provides us with about 4,000 of these. And in a previous study, I looked for these UCNEs across a large number of birds, including several parrots. And what I found was not only a suite of these ultra-conserved non-coding elements that were divergent specifically in parrots, no other birds. Interestingly, those UCNEs that were divergent in parrots were also clustered around specific genes. And those genes were involved in a lot of really interesting processes involving the brain, neuronal differentiation, forebrain development, projection neurons, as well as when mutated in humans, they seem to be involved in a number of cognitive disabilities, schizophrenia, intellectual disability, autism, different sorts of neurobehavioral variations like that in humans. Interestingly, many of these genes also had human non-coding regions that were accelerated relative to chimpanzees around them. So it's possible that these non-coding elements could be involved in the evolution of higher cognition, both in parrots and in humans. So this was a really interesting finding that then we wanted to extend to mannequins to try to identify, could there be similar ultra-conserved non-coding elements that are divergent only in mannequins and not in any other birds? So I took that same set of 4,000-ish UCNEs, the 5 Manakin genomes that we're focusing on, one closely related outgroup species, the willow flycatcher, as well as 12 other birds, sort of capturing the diversity of avian phylogeny as best we could. And what I found after narrowing down the set of UCNEs that were statistically divergent mannequins, removing the ones that were either problematic or that were not sufficiently divergent from that close outgroup species, the willow flycatcher, I identified 122 that were divergent in mannequins as a whole, as well as 57 that were specifically under selection in mannequins that perform elaborate courtship displays. So now taking a look at these, a closer look at these ultra-conserved non-coding elements that are specifically divergent in mannequins, they're not randomly distributed across the genome. As we saw with the parrots, they're clustered around specific genes, for instance, FOXP2. And interestingly, the genes that these UCNEs are clustered around, it's not just a random set of genes. In fact, it's genes that we might speculate could be involved in interesting Manakin behaviors. So one of the genes with the most Manakin divergent UCNEs around it was TLE4. And TLE4 we know is involved in visual processing. I'm going to follow up on that in just a second. FOXP2, which some of you might be familiar with as, the language gene. I think a more nuanced view of this gene is that it's not just involved in human language, but rather with a lot of aspects of motor learning, especially those that are mediated through the striatum. So this is really interesting from the perspective of this courtship display, that it potentially has some learned components to it. Another gene with a lot of UCNEs around it is NR2F1, which we know is involved in steroid hormone receptor signaling. Again, as you might have heard today, this courtship display it's very much a sexually dimorphic behavior, where the males are generally the ones performing these elaborate displays while the females are, observing and judging them. And so it's really interesting that we see steroid hormone receptors, a gene that regulates them, also coming up as having a lot of these Manakin divergent regulatory regions around it. TCF4, another gene that came up in this screen, is involved in muscle patterning. And of course, as I mentioned earlier, we know that Manakins have a lot of specializations in their muscles, not seen in songbirds. BCNN2 is another gene that we know from humans all the way to fish is involved in laying down pigmentation in specific patterns. And of course, as you've seen from their beautiful pictures, these Manakins generally have very vibrant coloration patterns. So perhaps these are regulatory regions involved in some of that as well. And finally, NR4A2 is another gene that came up as having a number of specializations in terms of the regulatory regions around it. And this seems to be a gene involved in the development of the dopaminergic system. So again, this is a really interesting set of genes to come up as having specialized ultra-conserved non-coding elements around them. So this is great in terms of the computational prediction, but then we wanted to follow up on it a little bit to try to experimentally validate it as best we could. And we did this using in situ hybridizations for some of these genes to see if they have differential patterns of expression between Manakins and non-Manakins. And of course, while the zebrafinch is a common laboratory study system, obtaining Manakin brains was a little more challenging. But luckily, I was able to, several years ago, take a trip to the Brazilian Amazon with this mobile field neurogenomics setup that I devised in order to collect gene brains that are of sufficient quality to do things like in situ hybridizations. And by collaborating with a very talented grad student in the Mello Lab, Alex Neviu, he was able to run these in situ hybridizations for us for genes of interest, both in the Manakin and in the zebrafinch side by side in some of these brain regions of interest. And what we found is very interesting. We looked specifically in the tectum because two of these genes that had a number of divergent UCNEs around them, TLE4 and MICE2, not only are they known to interact with each other, they're specifically known to interact with each other in the development of the tectum. And the tectum is a brain region that's involved in fast visual tracking, which of course is important not only for the birds, the Manakins, to be able to perform these high-speed courtship dances, but also for the females to be able to track and evaluate them in their performance. So what we saw by doing these in situs in Manakins and in zebrafinches is a reduced expression of these genes in the tectum. So again, you have divergence in these regulatory elements around this gene. And what we see is in both of these genes, there's now reduced expression in the tectum. Is this, a causal smoking gun? Not yet, not necessarily, but I think it's a really interesting indication that these computational predictions might not all be smoke and mirrors, because we see that the gene that we predict to be differentially regulated in a specific brain region, in fact, is. So that was really nice to see. So where I would love to go from here is actually collecting some primary epigenomic data in some of these brain regions of interest that we hypothesize could be involved in Manakin courtship behavior. One option would be to collect essentially what is a marker of a histone modification that we know is involved in enhancer gene regulation. This H3K27 acetylase chromatin immunoprecipitation sequencing. Another technique that I've been using in a number of species is ATAC-seq, which is a way to identify all the regions of the genome in a specific tissue or cell type that are open and available for transcription factor binding, which is, of course, the mechanism that mediates gene regulation. And this is, in fact, something that I've done in a number of different species, from macaque to mouse and rat and bats, is to collect these different brain regions and non-brain tissues and identify, using ATAC-seq, all of the regions of the genome that are open and available for binding and gene regulation in those species, in those specific cell types. And what we see is that there is quite a bit of variation, and I was able to identify a number of gene regulatory regions that are species specific. And perhaps more pertinently, with the collaboration of Fending Lab member Irene Capello, we were able to develop machine learning algorithms that can predict, just from sequence alone, what parts of the genome might be regulatory enhancers in specific tissues and cell types. And so what we're working on now with the collaboration of the Jarvis Lab is to collect zebrafinch tissues in some of these analogous brain regions to now extend these models to birds. And hopefully in the future, I'd love to collect primary brain tissue from manakins as well. Then we can generate these models in order to computationally predict what regions of the genome might be involved in the evolution of tissue-specific expression that could underlie this courtship display, as well as many other amazing regulatory innovations that are necessary to build a Manakin. And so that's amazing, but again, at that point, it's still a computational prediction. So how can we experimentally test and validate some of these predictions? So one additional technique that I've been developing is called the Global Massively Parallel Reporter Assay. So classically, a reporter assay, as I showed you in one of the earliest slides here, is a way of attaching a putative regulatory element to something like LAC-Z or GFP in order to see where, what tissues that regulatory element is active in. So another way we can do this is we can take all of these regulatory regions from different species that we predict to be active in one brain region or another, or in one behavioral context or another. And instead of just testing one at a time, like with a traditional reporter assay, we can take hundreds and even thousands of them. And instead of attaching them to something like GFP, we attach them to a random sequence barcode in a plasmid. And we can then test thousands of these in parallel injecting them into a mouse or a zebrafinch, and then look at whether the different species variants lead to differences in the expression of these barcodes in different tissues. This can tell us whether some of these regulatory elements have sequence variations that give rise to differential gene expression in these various tissues. And this, of course, is iterative. It allows us to improve our predictions through these validations, and ultimately allows us to build a much more nuanced model of how these phenotypes could be evolving. What are the most critical sequence features that allow for this tissue-specific expression that leads to the evolution of a novel behavior like Manakin courtship displays? So with that, I'll thank you very much for your time and attention. And of course, I'd love to thank the members of the Fenning Lab and my PI for supporting me in this work. Our many collaborators in the non-Manakin world that have helped with some of these experiments and access to genomes, as well as, of course, the many fantastic collaborators in the Manakin RCN family. And with that, I will open it up for questions.